Okay, well, so I have an exciting day because I have about, I think, 3 papers that I want to go over, but I'll split them up into different videos. Let me get this all organized. So, this first one that I want to go over is called Detection Dogs as a Help in the Detection of COVID-19. Can the dogs alert on COVID-19 positive persons by sniffing axillary sweat samples? And this is a proof of concept study. And keep in mind, a lot of these people are French speaking, so the verbiage isn't necessarily completely natural, but we can definitely still follow it along with it, and hopefully, and keep in mind this is also a preprint, so it's not fully gone through the peer review process, but it was just explicitly a proof of concept study to see if this is possible. And since they had concluded that it is possible, larger studies can be conducted. So we're moving up in the ladder as far as uh, like how good of a how would I phrase that? They're moving up in the ladder as far as what needs to be done to get this going from a proof of concept all the way to a possible medical technique. So what would be one of the benefits of having a dog be able to sniff out um, sick patients? So we know that this happens already for certain types of cancers, and we can train dogs the same way that we train any type of artificial intelligence, where we will give them samples, and then if they correctly guess what the sample is, uh, or what category that sample should be in, then we apply positive reinforcement, which then helps train the dog or sometimes like software or an artificial intelligence. And after doing that enough times, you can start predicting whether or not um, you can start predicting samples that had not been that have unknown labels to them. So this could be beneficial for, say, if you have a bunch of people that are walking out and about, and you obviously cannot run PCR tests on all these people. So what would be beneficial is if, say, someone were to contract COVID-19, even if it was a mild case, then is it possible that their biochemistry or their metabolism gets shifted in a way which ends up producing different types of compounds in their sweat? If that is the case, then we can actually utilize those that shift in metabolites as a way to diagnose, or at least, yeah, as a way to diagnose whether or not someone likely has COVID-19 or not. So, anyways, we will go into it, and keep in mind a lot of people are working on the study, and it's in multiple countries as well, so it's pretty robust, and it's not peer reviewed yet, but hopefully it ends up becoming that in the future. So I'm going to turn the fan on because it's a little toasty in here. So the abstract. The aim of this study is to evaluate if the sweat produced by COVID-19 persons, so SARS-CoV-2 PCR positive, so PCR, that's when we check for the genetic material of the virus itself. Okay, so this would be different to, say, an antibody test. So this is testing for uh, virus virus genetics still in the person's sample. And so evaluate if the sweat produced by an infected person has a different odor for trained detection dogs than the sweat produced by non-COVID-19 persons. The study was conducted on 3 different sites following the same protocol procedures and involved a total of 18 dogs. A total of 198 armpit sweat samples were obtained from different hospitals. For each involved dog, the acquisition of the specific odor of COVID-19 sweat samples required from 1 to 4 hours. So for each involved dog, the acquisition of the specific odor required 1 to 4 hours, with an amount of positive samples sniffing ranging from 4 to 10. So that is quite remarkable that it only takes between 1 to 4 hours to train these dogs to learn what scent is associated with COVID-19, and really it only takes 4 to 10 different samples, is according to this abstract. Maybe I'm uh, misreading this, but yeah, that is ridiculous. When we train For instance, when we train artificial intelligence or when we train neural networks to do classification, sometimes it requires hundreds, if thousands, tens of thousands of samples and can require hours upon hours of work to do that. Granted, this is a simple category 1 or category 2 classification, but still, it's quite remarkable that we can get these, that we can possibly get these results from just training a dog. Anyways, for this proof of concept, we kept 8 dogs of the initial group, the explosive detection dogs and the colon cancer detection dogs. So only these 2 dogs were these 2 subsets of dogs were used, and they performed a total of 368 trials, and they will include the other dogs in our future studies as their adaptation to sampling scenting takes more time. So keep in mind, these dogs are trained to use their noses, use their olfactory system to do some sort of job. And so this group wanted to take dogs that have already been trained to do sampling or to basically seek out specific scents. They took some of those dogs for this study, and some dogs are trained to do different jobs, and some jobs were easier to make the shift from their job to this COVID-19 sampling. So that is why they are only including 8 dogs from this group of 18 dogs. So they're only including these 8. Anyways, the percentage of success of the dogs to find the positive samples in a line containing several other negative samples, or mocks, 2 to 6, were 100p100 for 4 dogs. So I believe this means 100% for 4 dogs, and respectively 83% and 84%, 90%, and 94% for the others. So all of those significantly differ from the percentage of success that would be obtained by chance alone. So chance alone would be a far lower number, but these dogs seem to be doing a pretty good job. Anyways, we conclude that there is very high evidence, or that there is high evidence that armpit sweat odor of COVID-19 positive persons or people is different, and that dogs can detect a person infected by SARS-CoV-2 virus. So keep in mind, though, this is of people who have the disease in them at that moment. What I think would be interesting is to see how long their sweat maintains this odor shift. So if you can imagine, this would be important in, say, if we have a lot of people trying to reintroduce themselves back into society after quarantining. Let's say we trained a bunch of dogs to detect COVID-19, well, it'd be very annoying if those dogs were mixing up people who still have the active virus and people who had it a month ago. So that would be another level of distinction or a level of discrimination that we would like to see in our training data. Anyways, I should also mention that training dogs is similar to training artificial intelligence. The data you put in is going to be the data you get out. So you really got to be mindful whenever you do these data collection and data training, batch training, that you are truly getting a representative sample from each of them. Anyways, because you could also imagine that there might be differences based on diet, which if you go from one country to another, there could be a very large diet change, which could cause a big shift in body odors. Anyways, canine olfactive detection has proven its efficacy in numerous situations, such as explosives, drugs, and banknotes. Banknotes, really? To wonder what that would be for, to see if they are fake or not? That'd be... Really? That'd be cool. Anyways, including for early diagnosis of human diseases, various cancers, alert of diabetic or epileptic people in immediate alarm of crisis. So you might have seen some, there are some dogs that are trained to basically be on very high alert, utilizing their olfactive or their nose to know when their owner is going to have a epileptic seizure or not. So it truly is remarkable um, what senses, if you have the sense, how your body can use that sense to rationalize the world around you. So, I mean, we don't have that incredible sense of smell, and therefore, since we don't have that sense of smell, we're not using our sense of smell to learn as much about our environment. But for dogs, their sense of smell is so keen that they gain a lot of understanding of the world around them just from their sense of smell. It'd be similar to say echolocation for, bats and dolphins. That's one of their senses, and that's how they can gain a big understanding of the world around them. Whereas we cannot do that. So we are at a huge disadvantage in that regard. So continuing, the nosai, uh, the noses. I don't know how to pronounce this because believe it's French. I think it just means nose, but let's look. Okay, well, let's look at etymology. Yeah, I guess it just means dogs or something like that. But anyways, the No Size project, conducted by the UMES, or the Unité de Médecine de, I am so sorry, I speak zero French. But this UMES at the Alfred School of Veterinary Medicine, which is in France, has been set up in order to develop the scientific approach of medical detection dogs. The occurrence of the COVID-19 pandemic led to the Noseye's team to start a multiple center study on the olfactive detection of SARS-CoV-2 positive people involving this Alfred School of Veterinary, or we'll just say this hospital that is in Paris, this hospital, and this hospital in Lebanon. So there are multiple countries are being recruited. South, and I guess this is also in France. Anyways, fighting such a viral outbreak requires a widespread testing, one of the key measures for tackling the pandemic. In June, yeah. So for instance, uh, testing is key for tracking and tracing. Testing is absolutely key, and for knowing whether or not you're infective, uh, tracking is key. Testing is key. So if we can find another way, another method to test, that can lead to more robustness in our strategies. So our strategies need to employ multiple methods to become much more robust. And so by increasing the repertoire uh, of testing capabilities, we are creating a more robust society against a pandemic. Anyways, in June 2020, facing a decline of COVID-19, it is possible to say that countries that have mastered their outbreak and were able to maintain the number of infected people low need to perform fewer tests to correctly monitor the outbreak than those countries where the virus has spread more widely. And for the same reasons, the timing of testing is also crucial. A high rate of testing will be way more effective to slow an outbreak if conducted earlier on. So I was screaming about this. Test early. We needed, and the United States CDC was not exactly um, perfect. Okay, sorry, got a test. Okay, for this reason, and because of the potential risk of second wave COVID-19 in the North Hemisphere, and facing the spread of the disease in the South Hemisphere countries, we decided to launch the No-Size COVID-19 assessment on May 1st. So, the proof of concept study is based on the preliminary assumption that dogs, with their highly advanced sense of smell, could be trained to discriminate COVID-19 positive patients from negative ones in relation to the excretion of specific catabolites. So what are catabolites? So whenever you have any metabolic pathways or any metabolism that you do, sometimes there are waste products, or yeah, I'll just call them waste products. That's not necessarily the best way to describe them, but uh, catabolites are basically metabolites that come from the breakdown of something else. And these breakdown products can be viewed as a chemical signature of the, or a ghost, if you will, of the chemicals that it came from. So if you have big shifts in your metabolism, you will also have big shifts in your catabolism and your catabolites. Anyways, these catabolites uh, are excreted, and they can be excreted multiple ways, but the sweat glands is one of the big ways that they are excreted. So we're hoping that the excretion of these catabolites, we might have absolutely no idea what these catabolites are, but they could possibly be changed in expression by the SARS-CoV-2 virus cellular actions or replication in the sweat. And we don't know what they are, but we're hoping that if we just throw it at a dog will be able to figure out which ones end up working. And likewise, we could probably do a same or a similar experiment. We could do a similar experiment with computers. So if we could also take these sweat samples and run them through a type of like an artificial nose. I'll call it an artificial nose, but we can put it through, say, a mass spectrometer or something that analyzes the different chemicals that are in it. And by doing that, we can say, okay, this set is COVID positive and this set is COVID negative, and then we might be able to develop softwares or programs or artificial intelligence neural networks that are able to discriminate it. So when we bring it a new sample in, it can sense the chemicals in that sample, and then hopefully be able to properly diagnose the person that sample came from. So it's not just for dogs. We can do this for computers as well, but this is faster and this is easier to do because they're dogs and they're nice, they're fun to play with, right? Anyways, our first and main basic question though, does the sweat of a COVID-19 positive patient have a specific odor distinct from the one of COVID-19 negative people for trained detection dogs? So the medical detection dogs, the state of play. So here, volatile, I think they meant to say volatile, but they misspelled it twice. So volatile organic compounds, or VOCs. So these are, volatile means that they're going up, they're wafting, evaporating into the environment very easily. And organic compounds means that they contain carbon in it. And so these VOCs, you might, if you've done any work in construction or basically anything dealing with plastics or furniture that utilizes synthetic materials, frequently we talk about VOCs because they can influence health if you breathe them in. They can influence the environment if they can cause some sort of ecological damage. So there is a lot of research that goes into minimizing volatile organic compounds from our manufacturing process. But it's not just in manufacturing. You as an individual or any living organism really will be producing these volatile organic compounds, or these VOCs. So anyways, these VOCs represent a large range of stable chemical products, volatile at an ambient temperature, and are detectable. Maybe sometimes they're not, but like we presume that they are detectable for those of interest in dogs' olfaction in breath, urine, tears, saliva, feces, and sweat. So it's not just coming from the sweat. You could possibly do these VOC detections in a lot of other samples. And different samples might have different baselines as far as what compounds they release normally. Anyways, most studies on volatile biomarkers have been conducted on breath samples, even if other matrices, so urine and feces mainly, have been studied. Anyways, they are more and more frequently used for dog olfactive detection of explosive, drugs, banknotes, conservation, reintroduction and survey of wild species, endangered species traffic, and so on. So all of these, for instance, there are ways to hide. You can hide a lot of things, and you can visually occlude them so that visually you can't find these things. You can't find the problem. But if you have dogs that are trained to use their sense of smell, then they go from being unseen to being seen. So these are invisible enemies, right? Invisible enemies are very hard, they're very difficult to defeat. But if you can make the invisible visible, then all of a sudden you know where to attack. So explosive drugs, banknotes, uh, and then also the conservation, endangered species traffic, so on and so forth. These are all I should say, areas in which the trained dogs play a huge role in solving the issues of. So skin is the largest human organ, and it's accounting for approximately 12 to 15% of the body weight. Yeah, so they have a very interesting way to write percent here. Percent of the body weight. Volatile organic compounds emanating from the skin contribute to a person's body odor and may convey important information about metabolic processes. They are produced by eccrine, which is a type of what's it called, sweat gland, apocrine, and sebaceous gland secretions. I forgot what apocrine was. Relating to or denoting multicellular glands which release some of their cytoplasm, really, in their secretions, especially the sweat glands associated with hair follicles in the armpits and the pubic regions. Oh, it's kind of disgusting, actually. Wow. So basically, they can release some of their cytoplasm into the sweat glands. Anyways, and these are major sources of underarm odorants and play a role in chemical signaling. On this point, numerous studies have been performed in order to characterize the specificity of underarm VOCs produced by apocrine glands. So, tracking dogs trained to recognize an individual's scent on garments from a particular body, for example, chest, arm, underarm, or leg, are not reliably able to generalize the individual's odor or other body parts, suggesting that a different odorant milieu may characterize the back and the forearm. So they're saying if you train a dog, you need to standardize from which area you are collecting the sample from, because it's more difficult to generalize over the entire body than it is a single area. Anyway, sweat from the palms of hands, soles of feet, and forehead comes mainly from the eccrine glands and sebum. In this case, most of the compounds have been documented to be organic acids, usually carboxylic acids, ranging in carbon size from C2 to C12. So C2 would be acetic acid or vinegar, and C20. So these numbers indicate how many carbons are contained by that particular molecule. So a C20 would be an organic acid that has 20 carbons associated with it. Anyways, the most abundant being C16 and C18 saturated, monounsaturated, and diunsaturated, which are not volatile at body temperature. Okay? A more recent study confirmed that human sweat is different depending on the anatomic side on humans, and that the underarm sweat appears as the most efficient in terms of organic compounds turning volatile at ambient temperature close to body-like temperatures. So this is one reason why they're using the armpit rather than sweat from the palms or soles of feet or the forehead, because those are mainly coming from the eccrine gland and sebum. And so they contain a lot larger carbon compounds. C16, C18, those are the most abundant. And so these are going to be fatty acids, saturated, monounsaturated, and diunsaturated. And they're large enough that they're not really that volatile. They don't go into the atmosphere easily. Whereas you would normally want smaller carbon compounds because they are more likely to release into the air for a dog to detect. So this partly explains that in our detection dog study, we chose to sample underarm sweat in the armpits. So B, what? Non-infectious diseases detection dogs. So for centuries, our human sense of smell has been used by medicine practitioners, be it for the recognizing gas gangrene on the battlefield or diabetic ketoacidosis. Uh, so apparently, if you have gangrene, then you release a gas? Really? That produce gas gangrene that produces tissue gas and gangrene. This deadly form of gangrene usually is caused by Clostridium. About 100 cases of gas gangrene are reported yearly in the United States. Well, that is disgusting. So, yeah, apparently in the battlefield we can. Oh, the human sense of smell, so not dogs. So we alone can detect gas gangrene, and we can detect diabetic ketoacidosis, which ketoacidosis is when your body starts making a lot of ketone bodies due to the imbalance of your blood sugar. So improper carbohydrate metabolism can cause the production of ketones, and these ketones are very, very volatile. So you can start smelling them in people's sweat and their breath, which is, I would not consider it a pleasant odor, but to each their own. Regardless, scent detection by animals has been published in a large number of diagnostic studies, which all suggest similar or even superior accuracy compared with standard diagnostic methods. So every diagnostic method we have is imperfect. Biology is a noisy field, and therefore even our best diagnostic tests can sometimes fail, either give us false positives or false negatives. But even so, sometimes dogs or animal detection appears to sometimes be superior to our tried and true methods of what's it called? Sometimes they're even better than our other methods. And I should mention that in this particular paper, they discovered that some samples were actually improperly labeled because of the superiority of the dog's detection, or the dog's ability to detect positive samples. So that was, we'll get to that bridge when we come to it. Anyways, a bibliography request of PubMed, including the keywords dog detection and cancer, leads to 2,612 publications, demonstrating that the dog's nose is now considered as a tool for early detection of main cancers and their prevention. An exhaustive review was published by Engel in 2016 on the subject. The hypothesis that dogs could be able to detect malignant tumors based on their specific odor was put forward by Williams in 1989. The first clinical investigation of cancer was published by Willis on bladder cancer in 2010, after having published a proof of principle study in 2004. Several positive studies have been performed on colorectal cancer screening with odor materials by canine scent detection, and some tendencies have been recorded for lung cancer, melanoma, prostate, or liver cancers. So focusing on the non-infectious diseases, and mostly those are mostly cancers. We have been using dogs effectively for decades as a means to properly diagnose people for a specific cancer. So what does this tell me? It's telling me that if people get cancer, it's probably seriously affecting their metabolism in some way, and then we can see the catabolites or the metabolites that are associated with that shift in metabolism, and we might not necessarily know what chemicals they are, but if you start training dogs, yes or no, over and over again, eventually you can start getting good data, or getting good predictability in your samples, or in your model, I should say. So we do this, and we're starting to do this with computers now. Computers looking at, if we have computers that are looking at scans of people or looking at tissue samples, uh, now the programs are getting so good at detection of disease that they're starting to approach being just as good as the physicians themselves. So there really is a lot of room, or how to phrase that, there's a lot of good that can be done from these simple classification problems. So if you can turn your problem into a simple classification problem, you can learn how to train a software or a dog or any other semi-conscious or conscious entity to really be effective in diagnosing problems in the medical field. Anyways, I'm rambling. New diseases are starting to be explored. So, for instance, ovarian cancers or degenerative diseases like Parkinson. So that would be great if we can start doing early detection of Parkinson's based on the shift in metabolism of that person. Sorry. As dogs, training techniques are now much better known, and specific volatile organic compounds considered as solid biomarkers. So let me. Okay. Anyways, infectious and parasitic diseases. So several studies have used dogs in an attempt to differentiate a range of target odors among insects and parasites. For example, these people trained dogs to locate egg masters. I don't know what that, of the gypsy moth. Let me, yeah, that's not, I'm assuming, I don't know what an egg master is, but I would assume that it's just a group of eggs. I won't be able to tell you what that is explicitly. Regardless, a study conducted by these people assessed that the dog ability to differentiate between nematodes, infected and uninfected sheep feces. So give them the crap of sheep. A dog will be able to detect whether or not there's nematodes in that, and that could warn you to give an anti-parasitic to that particular sheep, which is great for the sheep. But recently, Guest did show that trained dogs can identify people infested. Guess that might technically be the right word, but infected with malaria parasites thanks to their olfactive capacities. So that would be, I mean, we don't need this in the United States nearly as much because malaria is not as widespread, but I could definitely see the huge social benefit to being able to train dogs to detect malaria in communities around the world, because then you don't need to have a blood sample done, and a lot of energy and time and money, ironically, is going into developing low-cost ways of detection of malaria. But if you could just have a dog sniff you and then tell you whether or not you have it, then yeah, that'd be phenomenal, because dogs are all over the world. So if you could train them to do that, wow, that would. You just need to feed it. You just need to feed the dog, and it can. It'll be a lot cheaper. Anyways, bacteriological diseases can also be detected by the dog's superior olfactory system, as Boehmers demonstrated during a hospital outbreak of Clostridium difficile. Rats, and this is C. diff, if I remember, which is a pretty awful intestinal disease. Rats olfaction is also used now for the detection of tuberculosis in East Africa, and that, I did not know that. Wow. In a study by Maurer, dogs were trained to distinguish urine samples that were positive for bacteria from those of culture-negative controls. So here we can see dogs being tested for not just one disease, but 4 diseases. And I don't know if they were training a different dog for each of them, or if they said, in this group, they are positive for at least one of them, and then in the negative group, it was all negative, which, you know, that would mean that there would be a common change in metabolism among all types of bacterial infections. And granted, you know, the more complex you make one group, the more likely you will have bacteria or organisms that are exceptions to the rules that have found ways to avoid detection. So it is possible that if you were to make this list too large, then all of a sudden there would really be no or very few common factors between them. So the more complex it is, the less common factors, and therefore the more difficult it might be to have proper detection, sensitivity, and specificity. Regardless, for those of culture-negative controls, and all dogs performed with similar accuracy. Overall, sensitivity was at or near 100%, and specificity was above 90%. So almost no false negatives, almost none, and very few false positives. Anyways, concerning virus detection, dogs are able to differentiate cellular cultures infected by the virus of the bovine mucosae disease from non-infected cultures, or from those infected by other viruses. So bovine herpes or bovine parainfluenza. So it is possible to, if you train the dogs correctly, that they can distinguish between specific viruses. So this is beneficial, especially if you wanted to differentiate between, say, COVID-19 and a different coronavirus, for instance. Anyways, the materials and methods. So the study conducted on 3 different sites following the same protocol. So Paris, I don't know how to pronounce it, and Beirut. And the samples were collected from the armpit, and those already explained in a previous paragraph of this paper. The sweat, and they chose the sweat because it doesn't seem to be a route of excreting the SARS-CoV-2 virus, and anticipating possible future practical applications of dogs trained to detect people carrying the SARS-CoV-2 virus. If the results of our studies are positive, sweat, even if not the only one, is the key odor for search and rescue or tracking dogs. And anatomical sites of sampling are armpits, considering the site is hardly passively contaminated by the positive patient. As the site is hardly passively contaminatable by the positive patient. Okay. Anyways, inclusion and exclusion criteria. So positive samples are realized on patients showing clinical symptoms who were COVID-19 positive on RT-PCR or PCR tests for SARS-CoV-2. So their positive cases are only going to be ones that have tested positive for the genetic material of the virus. Okay? And patients were excluded from the study protocol if they had, and I should also mention that they needed to show clinical symptoms. So this is for the mild to severe cases of COVID-19. It would not necessarily, it might still work, but it wouldn't necessarily work on a completely asymptomatic case, even though we do know that half of asymptomatic people seem to show some X-ray lesions in their lungs. So asymptomatic is a broad term. Is what I'm trying to get at. Anyways, patients were excluded from the study protocol if they had received a medical treatment for more than 36 hours. And they did this in order to avoid possible interference with long-term medical treatments in the sweat, volatile organic compounds. So remember, the medications that you're giving to people or the treatments that you're giving to people can seriously alter their metabolism. They can alter what products are being formed. The drugs themselves might break down, and those breakdown products might be somewhat volatile, and then that might be something that could confuse the dog. So the dogs might start smelling this drug catabolite, and then they might associate that drug metabolite with COVID-19 when really that correlation is just because of the treatment. They want to positively identify people without without any treatment. Anyways, samples are collected by doctors, interns, or nurses who are trained to not contaminate the samples with their own odors. A training video and the necessary equipment has been provided to each staff in charge of sample collection. Negative samples are collected the same way by the same trained staff from patients who met the following inclusion criteria. So no clinical symptoms related to COVID-19, and they are negative on the SARS-CoV-2 PCR test. So you can't, they wanted to make sure absolutely that no clinical symptoms for COVID-19 were shown, and that they were absolutely negative for SARS-CoV-2, at least on the PCR test. That might not necessarily be the case, as we will see later. So this is what I found very promising, was it seems as though whatever method was used to train the dogs, whatever chemicals they latched onto to tell them that someone was COVID-19 or SARS-CoV-2 positive, it seemed as if that shift in metabolism is happening early on. It's happening early on in the disease progression, or it's happening with the asymptomatic cases as well. So anyways, in order to avoid any dog I don't know what that means. That might be yeah, I don't know. I don't know what this word is. In order to avoid any dog creation, or okay, we'll keep on reading. On the olfactive background noise of positive sampling sites, both positive and negative sample collections were made in the same location. Okay. Yeah, try to avoid background noise. Get both positive and negative samples from the same location. Continuing, total of 198 samples, 101 positive samples and 97 negative samples were collected at the endpoint date of the present study, and their distribution according to the sites and sex is reported on Table 1. So sampling supports. So two types of sampling supports are used. Sterile gauze swab utilized by the hospitals, or sterile gauze filters utilized for explosive detection, and inert polymer tubes used for explosives, drugs, or criminology detection, and this is provided by the Gextent company. The polymer tubes, and this is important, the polymer tubes are able to adsorb both polar and apolar molecules. So whatever they are sampling, some of those chemicals will stick to the surface of the polymer tube, and then that polymer tube will be sent to the University of Corsica Pascal Paoli, and then they're going to do a chemical analysis on those samples to see if, so for the dogs, they're just saying, dogs smell this, and if you smell something weird, let us know. But what we are going to do later is do an actual test on what precise chemicals are in it, and that might also teach us, are these the chemicals that the dogs are smelling? And then furthermore, it can tell us, oh, so these chemicals are being secreted at a higher or lower rate if someone is COVID-19 positive. And so there, we might be able to use those to gain some insight on how your metabolism is being screwed up by the COVID-19 disease. Anyways, here are the sampling techniques. This is how they are sampling. So the patient hand is holding that back, and they're sampling the armpit. And then these are the polymer tubes that they're sticking everything in, and these are the cylinder filters. Let's see. Samples are put into an so samples are put into a UV-resistant sterile container, and the container is disinfected by the helper. It is coated, and then those are placed in the second plastic envelope. A sample is never used for dog training prior to 24 hours after sampling the sweat, as the SARS-CoV-2 virus does not survive more than a few hours on a cotton and disposable gauze. So they purposefully waited at least 24 hours so that if there was any viable virus on that in that sample, they would hope that it would die, and this would be rendered inactive over the 24-hour time period. And this would be beneficial, especially if you have dogs sniffing it, because we do not want to have a case where a dog ends up contracting SARS-CoV-2 if they happen to be unlucky enough for that. So a more recent study, concludes that absorbent materials like cotton and gauze are way safer than non-absorptive materials for protection from SARS-CoV-2 infection. So. All coated samples are then stored at a constant temperature of 18 degrees centigrade and hygrometry, so air humidity, I should say. Before being sampled, each person receives explanation on the protocol and signs and individual enlightened agreement, informed consent, form that stays by the hospital in the medical files of the patient. So here's all of this. Here's the form that they can read and sign. And then this is a table. So they wanted to also show the dogs as well, because I'm sure that if you are doing work with dogs, that you gain an appreciation for them, and you want to share your appreciation. So here are the dogs. So we just present two of the dogs who participated in our study. So here's Oslo, and here is Gun. So let me see if I'm missing anything. So the 18 dogs that started the experimentation and their characteristics are regrouped in Table 3, and they belong to the Parasite, Firefighters and Emergency Department 77 is where one of these dogs came from. This site, the Firefighters and Emergency Department 2A, they provided dogs, and then at the Beirut site, I don't know what this is, but this is the area or the organization that they got these dogs from. So remember, these are dogs that are trained for different jobs normally. For the study, we decided to include 3 different types of detection dogs. Here we have the explosives detection dogs, trained to detect 30 to 40 different types of explosives, but moreover used to work on a line of samples that they have to sniff individually. So for such dogs, if COVID-19 samples had a specific odor, they only have to upload one more odor in their olfactive memory bank. So for these dogs, they are specifically trained to detect a lot of different types of chemicals. So we would hope that it would be easy to train them to learn one more. And the search and rescue dogs are normally trained to perform in a disaster area search, and mainly work through the scent of sweat of the persons, which explains also our choice. And the colon cancer detection dogs trained on rectal. These dogs smell farts to find colon cancer. Did you know that dogs can be trained to smell your farts to see if you have colon cancer? That's great. Yeah, I bet they're really happy. I love this. Anyways, we did not decide to work with drug detection dogs, as there is always a possibility that COVID-19 positive or negative people use prohibited substances that would let catabolites be excreted by the auxiliary sweat. So, um, dog detection drugs would probably be very difficult to train for this particular study, because it is possible, it is actually very possible that they would have false positives, because they could be crossing their wires as far as what they're supposed to be looking for, and you cannot, you can't verify whether or not a person uses, uh, if a person has used illicit substances, weeks prior, because if especially if they're taking substances that might stick around in your body for a long period of time, you might still see those in the sweat as catabolites for a long period of time, which could theoretically confuse the dog. So you would probably not want to have a dog that was trained specifically for drugs to be in this test. Anyways, most of the dogs included in our study are Belgian Malinois shepherds. Is that how you pronounce it? Pronunciation. I wish that they would just, oh, here. I'm going on a tangent. I apologize. I just want, I'll look. If you speak French, can you tell me how to pronounce this, please? Anyways, because it is actually the most represented breed in working dogs, French structures, or I guess their companies or their infrastructure. Besides that, but we insist on the point that lots of canine breeds, or mongrel dogs, could develop the same olfactory detection qualities. So what is the official definition of mongrel? So it's just of no definable type or breed. So a mutt. Okay, well, could you imagine a pug being used for this study? That'd be great. Anyways, so here are the dogs. Here are their names, their gender, their breeds, their ages, they're calling it structures. I think a better English term might be what would the word be? Departments. So the departments that are involved, that'd probably be a better word. And here are the specialty. So they're typical jobs. Anyway, safety of the dogs regarding SARS-CoV-2 virus infection, and there have been very few isolated and criticizable reports on the passive carriage of SARS-CoV-2 virus by the dog with very small amounts of viral RNA, indicating that the samples collected by an infected person had very low viral load. The low viral titers or concentrations observed in the dog suggests that it developed a low productive infection, and the likelihood of infectious transmission was minimal or not existent. So there were some dogs that tested positive for SARS-CoV-2. Theoretically, SARS-CoV-2 isn't good enough at latching onto the dog's ACE2 receptor, so it wouldn't be nearly as pathogenic, and as a result wouldn't be as infectious, but we would rather be safe than sorry, and we would like to not give the virus the opportunity to, if it had mutated, to infect our dogs, our precious dogs. So this is one of the reasons why they're waiting 24 hours before they run the tests. Anyways, a second case was alerted in Hong Kong when the owner tested positive for COVID-19 infection and stayed in quarantine with his 2 dogs. One of them tested positive for quantitative PCR, but never had any symptoms, and the other stayed negative. As with the first dog, the infection was very low positive and non-contagious. So I don't know what to think about the Hong Kong ones. I don't know if it was this particular man, but there was a dog that tested, it was a Pomeranian, that tested positive, and then it died shortly after, but they said it was due to the pre-existing conditions of the dog and it wasn't related to COVID-19 at all. But the owner refused to allow the scientists to do an autopsy on the dog to see what exactly happened. So, yeah, I mean, I do have a little bit of concern as what will happen to some of our dogs as a result of this. Anyways, in the United States, IDEXX Laboratories tested more than 4,000 canine specimens during its validation of a new veterinary test system for the SARS-CoV-2 virus and found no positive animals. More recently, a first study conducted in the Alfred School of Veterinary Medicine showed the absence of SARS-CoV-2 infection in dogs in close contact of a cluster of COVID-19 patients. So finally, the CDC in the United States and ANSES in France attest that there is absolutely no evidence that pet animals, especially dogs, play any significant role in the transmission or in spreading the virus that causes COVID-19, and the risk is considered as close to zero. Well, especially since we don't usually let our pets intermingle with a lot of other people, then due to that, there's probably a very low transmission risk. If the dog is having close contact with the person, you're probably also having close contact with that person, and therefore the dog's probably not the predominant way at which, if you did catch COVID-19, it probably wasn't through your dog. And then also, there's a limited amount of there's a lot of social distancing between pets in general, because pets frequently are usually closer to their owners rather than with other members of their own species. Anyways, training and first testing of the dogs, and the dogs are first trained to work on a line of sample carriers, and the dogs are taught to sit in front of the cone containing the positive sample, and the method is based on a positive reinforcement. So the dog gets his toy for each correct marking. So this makes me feel warm inside. I like this test. So here they have the glass jar with the sample where they put the polymer tube, and here's the cone of olfaction, where the smelling tube, or the smelling cone, and they will line up 4 samples, and then once the training period was considered as completed by the dog's handler, the dog was not anymore in contact with any samples prior to a trials session, and the data collection regarding the success or failure to detect COVID-19 sweat odor started. And at the time, the dog must alert for COVID-19 positive sweat odor contained in 1 box out of 3, 4, 6, or 7 boxes, depending on the site study. So only when they line these up, only one of them will be COVID-19 positive, right? And then all the others will be negative. So the other boxes contain negative samples or mocks, but always with only mock in the line. Anyways, the whole period lasted 21 days because most dogs cannot work on a daily basis. Understandably. You gotta rest. So no disease symptoms or clinical abnormalities have been reported on any of the dogs involved in the study during the whole testing period. The standard environmental conditions for canine olfactory detection were observed. So, here we go. The dog finds the right one, and let me skip through this. So, basically, if the dog gets it right, they get the toy. If the dog incorrectly marks one, they don't get the toy. Anyways, results and statistical analyses. So, Since the objective of the study is a proof of concept one, we did not aim to provide evidence that a whole population of trained dogs are able to detect COVID-19 positive samples, but rather to provide evidence that a well-trained dog is able to detect COVID-19 positive samples. So such evidence is provided. So such evidence is provided if one observes one observation from one dog is a higher proportion of success than one expected by chance alone. Oh, okay. So such evidence is provided. So if one observes that one dog has a higher success rate than random chance, that is evidence. Therefore, the target population is not a population of dogs, but the whole population of trials, consisting of an infinite number of trials for one dog. So each dog is considered a diagnostic tool. They're saying, okay. Said, don't worry if it's not all dogs. Like, you just need to be able to train a subset of dogs. That's what they're getting at. And at the end point date, 3 search and rescue dogs have been withdrawn from the protocol for being unable to adapt to an olfactive search on a line of samples, and 8 of the same category are late in their testing period due to this necessary basic retraining. So let me So remember, can you teach an old dog new tricks? Well, sometimes you can, but it takes time. So they left. There are still a lot of dogs that have to go through this training process in order to even do this test. You need to be able to train a dog to do that first, right? So some of the dogs were not able to do this even in the first place, so they're still undergoing training in order to do it. But out of the 8 that were in it, there was some success. So the results are therefore presented on the 8 dogs who followed the whole process, as they appeared demonstrative enough to answer our first question. A total of 232 trials were made using 33 positive samples in France, and 136 trials for 68 positive samples in Lebanon. In order to provide evidence that the successes were not obtained by chance alone, the proportion of successes for one dog out of the total number of trials was calculated. Along with it, 95% confidence intervals calculated by using the Klopper-Pearson's method. The proportion of success obtained by chance alone was calculated by dividing 1 by the total number of sample carriers placed in the sniffing line. All the trials for one dog used the same total number of sample carriers. This total number of sample carriers depended on the site location. If the value of the random choice was not included within 95% of the estimated proportion of successes for one dog, the null hypothesis, according to which the dog chose the COVID-19 positive samples carriers by chance, was rejected with a probability type 1 error of 5% or 0.05. I forget the type errors. Regardless, they did a statistical analysis, and it was definitely statistically significant from random chance. So table 5 presents the results obtained from the 7 dogs of the study and the observed proportion of successes for each dog was significantly different from the one it'd be different from random chance, right? So here, is that his name? His name's Guess, right? Yeah, so number of trials. You know, the people probably name these dogs to be puns of their jobs sometimes. Anyways, number of trials, correct identification. So here, 84% success rate. Here's, I mean, you can look at this. So 4 of them, HK, Esco, Bella, and Jackie, these people got it 100% of the time. These dogs got it 100% of the time. And then obviously you need to do some statistical analyses, but you can say, yeah, it's pretty much 100% for 4 of them. So it doesn't matter if all dogs aren't able to learn this. You just need to train a subset of dogs. And here you can see these 8 well above, a lot of statistical significance away from random chance, which is good. It's good to see. And here is a graph of it, quite remarkable. So the results of this proof of concept study demonstrate that COVID-19 positive people produce an auxiliary sweat that has a different odor for the detection of dogs, and that than COVID-19 negative persons. It strongly suggests the hypothesis according to which dogs can be trained to detect SARS-CoV-2 contaminating people or contaminated people contained in auxiliary sweat, which is easy and safe to collect. So it's safer to collect sweat than it is to, say, collect throat swabs, right? Because they're opening their mouths, they're breathing, they might cough or sneeze while they inject when they put the swab inside of you. So this is a much safer method. Anyways, regardless of the nature of the dog's detection system and the fact that biology will never be an exact science, yeah, biology is noisy. It's never an exact science. So that's just one of the things that we have to account for. Both false positive and false negative errors occur in every single detection system, including highly sophisticated machines. So, you know, everyone makes mistakes, and even machines do. So just keep that in mind. So uncertain, a negative result of real RT-PCR regarding COVID-19 range from 22 to 30%, and RT-PCR results from several tests at different points appear variable from the same patients during the course for diagnosis and treatment of patients. And I really like this. In our proof of concept study, 2 negative samples, according to our described inclusion criteria for negative samples, which means that they did not show any symptoms and they tested negative, these people were marked positive by 2 dogs, which is remarkable. These people that tested negative were marked positive by 2 dogs, and so this information was relayed, sent back to the concerned hospital through the anonymous sample codes, and then they did more PCRs. That the PCRs were redone on the patients, and it gave positive results. Okay, so the dogs were better at the PCR. Now, granted, this could have meant that they just did not have a large enough viral load at the time to be detected by PCR. However, even with the small viral load, there was there was enough of a change in their metabolism, their metabolites, so that a dog could detect it. Or it could have been chance. But very unlikely for it to be chance. As said, the dogs are sniffing for specific molecules induced by the SARS-CoV-2 virus presence, and dogs have no reason to lie, as long as there is no environmental disturbance during their work. By registering all of the events, data, and filming every single trial for each dog, we were able to understand the reasons for some of the rare false negative markings that occurred. For instance, a horse passing... What? So apparently a horse passing closely by the working room, or the presence of a too zealous television team that did not respect the edited procedures for the sample. So yeah, they looked back at the videos and said, yeah, the false negatives. Wait. Yeah, so some of the false negatives where the dog picked the wrong area, they looked and they saw, yeah, there was an error in the way that the experiment was conducted. So that also lends credence to the original hypothesis. Anyway, sniffing behavior of the dog is actively controlled during investigatory behavior and rapidly modulated the response to sensory input so that the transport of volatile organic compounds through the olfactory epithelium and thus the olfactory proceeding is optimized. Another case of false positive marking in our study looks interesting to analyze. We took also negative samples of auxiliary sweat on some of the people directly involved in our study. We sampled ourselves, right? That's how you know you're doing good science. And one of these samples made on a young woman was marked positive by 2 male dogs without any environmental disturbance. But the dogs showed more excitation from the sample than usual for other positive ones. So we investigated the case and found that this woman had been sampled during her fertile period, and therefore we looked at sexual pheromones related to that period. So, yeah, well, this is very interesting. Okay, so here, one of the women who was one of the scientists on the team was at her peak she was peaking in fertility at the period of time of sampling. And that really got the dogs excited. That convinced the dogs that this woman had COVID-19. So that's interesting. Wonder what hormones are associated directly with the peak fertility. I'll look into that. So androsterone, androstenol, and isovaleric acid contribute to odor profiles, and the human axillary odor seem to have a communicative role analogous to that of the odors from specialized skin glands of animals. Some animal species have apocrine pseudoral or apocrine sebaceous glands similar to those of the human axilla. These glands, so there's some similarity between animals and humans, especially for the release of some chemicals. And anyways, these glands have prominent social functions, including a complex system of chemical messages, pheromones that provoke specific types of behavior. So published studies showed that the presence of androsterone in female axillary sweat makes females more sexually attractive in different species, including humans. So I actually hadn't read down this far before I decided to do a video of this. So here they're saying that the dogs were getting excited not because they detected COVID-19. They're saying they got excited because the woman is secreting fertility hormones, which would indicate to the dogs that she is in heat, and then would get the dogs excited, presumably. In Shakespeare's day, a woman in her fertile period used to hold a peeled apple under her arm until the fruit became saturated with her armpit scent. Then she presented this love apple to her lover to inhale in order to provoke his sexual excitation. Pheromones are defined as substances produced by animals which convey information to other individuals by olfactory means. And in such situations, androsterone and molecules like benzoate derivatives, so benzoate, you might have heard of like sodium benzoate, which is used as a preservative. These are things that look similar to benzoate. This is excreted in axillary sweat, enhance sexual attraction by men towards women near the end of the follicular phase of the menstrual cycle, and when fertility is at its highest. And in bitches, female dogs, during estrus, these molecules are part of the secretions that are highly attractive to male dogs and enhance sexual excitement. This fact explains the fail or the false positive of the 2 male dogs, and shows that such situations should be included in future male dog training on human axillary sweat samples. So these false positives were only seen in the male dogs, is what it sounds like. In our study, like in many others conducted on dog olfactive detection, the performance is defined in accordance with what is called the signal detection theory. Concha describes it as follows. True positive the dog indicates that the target odor by a sit response. False positive is the dog alerts to a non-target position, which is a control. A false negative is the dog fails to exhibit the trained alert in the presence of the target odor. And then the true negative is the dog does not alert in the absence of the target odor. All the trials um, Yeah, so in all the trials, the dogs were filmed in order to check afterwards more precisely their sniffing behavior. This will allow us to determine the duration of each trial before the dog alerts. The encoding of the presence or absence encoding stimulus is very rapid in the dog, and its discrimination seems to require only one sniff. Therefore, thinking about the future practical applications of our study, prolonged sniffing does not seem to be necessary when the target odor is absent. Anyways, future analysis of the results will also allow us to have a better appreciation of the impact of the results of the and the impact of the quality of the samples on the olfactive performances of the dogs. We will also compare the marking behavior of the dogs with the characteristics of the sampled COVID-19 positive persons. We will work on some limits we are aware of, like the eventual presence in the organism of the COVID-19 positive patients of other pathogens. So other pathogens could cause metabolic changes as well. The dog's attitude facing other types of respiratory diseases, and the correlation that might exist between the dog alert on positive samples and other biological tests that other than RT-PCR. Finally, after this working period, we also can conclude what will look at what is an evidence for good cynotechnicians. Dogs already trained to work on a line of samples and explosive detection dogs can be efficient to detect COVID-19 very quickly in only a few days. So these dogs that are already trained for certain applications, they can be retrained very quickly. Whereas search and rescue dogs need first to be readapted to a new methodology for them and will take a few weeks to be efficient. So we conclude that there is a high evidence that dogs can detect a person infected by a virus responsible for the COVID-19 disease. And in fact, even if they aren't showing symptoms for the disease, the dogs might actually be able to detect it. So let's go back to these lines. So I'm guessing that since these people do a lot of work on the nose, they are acutely attuned to anecdotes from history about how people used to use odors to try to elicit sexual arousal, right? So here, they're indicating that the dogs are creating these false positives due to sexual arousal. But there is another option, that during the follicular period, or the end of the follicular phase, that women might be releasing similar compounds that are associated with COVID-19. So let's look at the follicular phase hormone levels. Let's see. Let's do Wikipedia. Well, that might be it. So there was another. There was a study that was done on it was mostly men, but they studied the hormone levels of COVID-19 positive patients. And at least in men, it showed that their prolactin levels were extremely elevated and their luteinizing hormones were extremely elevated. And the luteinizing hormone is extremely elevated at the end of the follicular phase. So it could be that these sex hormones or these reproductive hormones are being modulated by COVID-19. I don't think that we should rule that out, because y'all might be aware that I'm on my high horse about how prolactin is probably being released by the presence of these virus particles that are mimicking the prolactin suppression hormones. So yeah, this is something to really consider. Does COVID-19 screw up your hormones in a way that is similar to the menstrual cycle, at least in this area of the menstrual cycle, when luteinizing hormone is highly increased? And yeah, in fact, let's look up, is prolactin prolactin menstrual cycle. Hyperlactin levels interfere with the normal production of other hormones, such as estrogen and progesterone. This can change or stop ovulation, the release of an egg from the ovary. It can also lead to irregular or missed periods, so that would be doing the opposite. With the highest level being during the ovulatory period. Let's look at this real quick just to see. Prolactin levels during the menstrual cycle. So highest level being either during the ovulatory period or during the luteal phase. However, the mean level of prolactin was significantly higher during the ovulatory and luteal phases than during the follicular phases. The mean level of prolactin, so this is the average. A direct relationship between oestradiol and prolactin levels was noted, although there was no correlation between prolactin on the one hand and follicle-stimulating hormone, luteinizing hormone, and progesterone on the other. So let's go back to this. And then hormone level, so this is ovulation right here. So this is when prolactin levels would spike. So this is something that really needs to be looked at, because this is where both luteinizing hormone and prolactin would go up in this area. And we do know, and prolactin is actually, I believe it's released in order to get the luteinizing hormone down, as you see here. So prolactin is probably being released, somewhere in here. I don't quote me on this. I would have to look at the actual cycles. So if you could imagine, if someone has both high luteinizing hormone and high prolactin, they're probably emulating this area, this ovulation area, and whatever hormones are associated with that. And knowing that's the case, a woman who was in this part in her cycle would also have similar metabolites. I think I'm probably gonna write an email to the authors of this, because yeah, that seems like an important fact that they should know, that some people have extremely high levels of follicle stimulating, or sorry, luteinizing hormone and prolactin if they are COVID-19 positive. And yeah, I think they should know about that because they're writing it off as if the dogs are getting excited um, about a fertile mammal nearby. So that's why they're going to the that's why they're doing the false positives. However, it could actually be the case that a woman who is right at this area of ovulation, or at least approaching ovulation, they are going to be not only in their most fertile area, but their metabolome, their their catabolites that they release are probably going to be similar. And so that might be what the dogs are looking for in the first place. Interesting. I I will look into this, and I have like two other papers that I want to record today. But yeah, we'll just try to rush through this. So, anyways, I will leave. Um, and okay, you can read this if you want, but it'd be a cheap tool. Uh, I will leave the reference to this in the description if you want to go see it or read it yourself. And thank you for listening, and I'll try to come up with another video later. So yeah, and I'm thinking also I'm probably going to do the like short summary videos where I go through. I'll call them recaps. I will recap the papers that I have gone over. That way, I don't have like a 2-hour long video that people have to go through. It could just be like a 2 or 3 minute get it done with. And if they are interested in actually seeing the full 2-hour video, then it's a free world. They can do it. Anyways, thank you for listening, and hopefully I will see you in a few hours.